Before I begin, I am truly honored to be here, and I thank Arijit and Mayappan for inviting me. And I also thank the student volunteers because we have made all the efforts for making our stay comfortable and making all the arrangements for all the participants. So today I'll be talking about machine learning aided discovery of potential biomarkers for cancer diagnosis and prognosis. Basically, I'll be covering a brief about the latest papers, and then I will move over quickly to our own work in this area, which has been also recently published. So before that, I belong to ICGEB in New Delhi, and I am heading the translational bioinformatics group at ICGEB, and basically we use various bioinformatics tools in solving various problems in health, agriculture, and biotechnology. So, but today my focus of my talk will be on our recent research on cancer. So, cancer ranks as the leading cause of death and is an important barrier to increase life expectancy, as you know. And worldwide, an estimated 19.3 billion new cases and around 10 billion cancer deaths occur. That is 1 in 6 deaths which have occurred in 2020 were attributable to cancer. And according to estimate from WHO in 2019, cancer is the first or second leading cause of death before the age of 70 years in 112 out of 183 countries and ranks third or fourth in a further 23 countries. So you can imagine the burden of cancer that has been seen worldwide. And especially in Asia, we see that a major chunk of cancer cases are reported in Asia also. So, of all the cancers, lung cancer accounts for second most number of cases, where it accounts for the most number of cancer-related deaths globally. So, although the number of cases are less, but number of deaths due to cancer, lung cancer are more. That is around 18%, whereas number of total cancer is lung cancer are 11.4%. So you can imagine the burden of lung cancer amongst all the cancer or amongst the whole worldwide population. And in India also, recently there is a report from ICMR that nearly 50% of the lung cancer in men and 55.5% in women are diagnosed when cancer has spread to other organs. So that is, it is detected very late, because of obvious reasons of not availability of secondary healthcare throughout the India uniformly. So it's very important to have proper early diagnostic tools which are accessible to population as well. So there is a lot of importance of accurate diagnosis and prognosis for precision for cancer patients. So accurate diagnosis and prognosis are instrumental in determining the most effective treatment plans for cancer patients, and it directly impacts patient outcomes and survival rates, making it a vital element in cancer care. So it's very important to diagnose as early as possible and also diagnose what particular stage of cancer a patient is in order to provide a customized treatment regimen for these patients. An effective diagnosis and prognosis influences medical decision-making for cancer treatments and interventions. So since cancer is a disease of genes, a lot of importance is there for sequencing cancer genomes. And we have also seen that there is a tremendous growth of cancer-related DNA sequencing sets in last 1 or 2 decades. And the reasons are that there have been tremendous advances in next-generation sequencing and other high-throughput techniques. And affordability is also another factor which has made availability of these techniques to different parts of the world. You can see here that when the first human genome was sequenced, the cost of sequencing was too high, millions of dollars, and then recently it has come down as low as $1,000 to sequence a complete human genome. And we can also see that several projects which were not possible earlier, for example, 1,000 genome projects or Genome India project, which were not possible earlier, are now possible to be undertaken. And 1,000 genome projects produce 2 times more data submitted to NCBI in just 6 months than that was deposited in last 30 years. So you can imagine the amount of data that is being generated by sequencing different genomes. And mainly it is helping the diseases of the genes, especially cancer. And recently we have seen that biological data storage is now more expensive than actually generating it. For example, if you want to archive data of population, make it private, make it anonymized and so on. So cost of keeping such a data set is tremendously very high as compared to actually generating it. However, there are various benefits and challenges when you are sequencing data. That you can learn from data to gain knowledge from experience and to make predictions which were impossible earlier. So you can now work on many problems which were not imaginable earlier. In future, real-time transcriptomic, metabolomic, proteomic and epigenetic profiles of individuals will be used for better personalized diagnostics, prognostics and treatment plans, which was not possible earlier. So this is just a snapshot. I'm sure most of you must have seen that it shows a tremendous growth of genome sequences that have been deposited to publicly available resources with the advancement of various sequencing techniques. This is the point where 1,000 genome project was accomplished, and then several other projects like TCGA, etc. were undertaken. And these colored lines actually show the capacity of sequencing the genome in coming years. So this was plotted probably in 2016, and now it was imagined that the capacity to sequence will go up to 1 zettabyte base pairs. So this is about various DNA sequencing projects which have been undertaken and the amount of data they are generating. However, there are other types of also high throughput techniques which are also generating a lot of data, which needs to be integrated along with the DNA sequencing data. So the DNA sequencing data alone will not be useful when you are talking about getting fruitful results from all these high throughput experiments and projects. So there is a need for integrative omics analysis, and I think somebody else also spoke about this probably yesterday, that it is important to have a bird's eye view of all the omic techniques in order to identify the real knowledge from various omic techniques. So luckily, machine learning has found a lot of use in application of analysis of various omics data sets in coming years. So these are artificial intelligence. Machine learning is a subset of artificial intelligence. Deep learning is a subset of machine learning. And they are playing probably potentially a very important role in analysis of various kinds of data sets, which will allow us to make advances in precision oncology, drug discovery, imaging, digital pathology, analysis of next generation sequencing data, especially for cancers, patient data management, even after they are admitted into hospitals. So there are various research which are being targeted towards exploring and exploiting the potential of machine learning in various cancer data sets. So recently, these are snapshots of few papers that people are using machine learning for classification of CT scans or histopathological slides, or even gene expression data sets and other data sets to unravel the potential of machine learning in solving some of the challenging problems in cancer data set classification, which are very important for personalized care in cancer. So these are some of our papers which we have targeted towards analysis of cancer data sets, and several other projects are also underway in the lab, including image classification based on histopathological images. So today I'll target my talk on machine learning analysis of lung squamous cell carcinoma gene expression data sets, which have enabled us to reveal novel gene panel which can be of potential prognostic value. So lung cancers can be classified broadly into small cell lung cancer and non-small cell lung cancer. Uh, Small cell lung cancer constitutes around 10 to 15% of all cancers, and non-small cell lung cancers, LUAD or lung adenocarcinoma, constitutes around 40% of all NSCLC, whereas LUSC constitutes around 25 to 30%. And uh, we decided to work on LUSC for certain regions. And we based our study rationale on the following hypothesis: that we can use machine learning to analyze LUSC gene expression profiles and compare it with survival analysis of patients from the same patient in which we are using the gene expression data in order to make some prognostic signatures. So this is the study workflow where we retrieved around 12 microarray gene expression data sets from NCBI GEO, and then we integrated all the data sets, and then we removed batch effects after merging the data sets because they were from different experiments and different samples. And then we actually adopted two approaches. One was we merged the whole data set, and then in second data set, a subset of the whole data set was made by extracting only the driver genes. So we took out genes which were labeled as driver genes from three different driver gene data sets called Intogen, Belly, and the one. And then we made a subset of these. So there were around 919 in the driver gene data set and around 20,000 in the complete data set. So then we partitioned the data set into training and testing, and then we perform feature selection from the partition data set in order to get the most valuable features which can be used as for efficient classification of the machine learning model. And then we also perform differentially expressed gene analysis of the data sets, and then we took out the intersecting genes, and we also performed further genes feature selection using lesser penalized Cox regression analysis, and which allowed us to further shortlist the number of genes to work with, which are the most probable genes which can be of any prognostic value. So I will show you a couple of snapshots now. So this is all the 12 gene sets that we used. So they were representing the cancer patient versus the normal, and some of them also had overall survival analysis data along with it, which can be used for making a risk model later on. So, and we also kept aside a certain GEO data sets for independent data validation. So these were used for training. So there were around 530 for LUSC and 430 for normal patients. And validation was carried out on 164. So this is plot just to show the batch effect removal using PC analysis. And machine learning, again, I will summarize that we used a two-pronged approach where we used a merged data set and a data set consisting only of driver genes. Then we performed feature selection, and then after partitioning into training and testing, we performed machine learning using 5 different algorithms. And notably here in the feature selection, we used 10, 20, 30, 40, 50, and 60 features in order to train all the 5 algorithms and optimize them. So here are the results from the best machine learning models that we were able to train. For internal validation, we found that the SGBoost classifier performed the best, and in precision recall also SGBoost and random forest algorithm performed the best. And for the driver genes where we used only around 50 features. So for complete gene, 40 feature data set was good enough to come up with the most efficient model. And for driver genes, the top 50 features, when trained on XGBoost, were able to perform the best. Similarly, when we did external validation, we found that the best performance was given by KNN, and in the second, in the precision recall also, this KNN was the best, and also in the driver gene data set with training on 50 features or 50 genes, again KNN was the best. And this is the overall accuracy for the machine learning model that we found to be the best. So we found that KNN is the best one amongst complete data set as well as driver data set trained on 40 and 50 features respectively. Then we performed a differential expression analysis, and we also validated our differential gene expression by partitioning the whole data set into random sets and then calculating the differential gene expression again, and we can see that the overlap coefficient was fairly robust. And this is just to show the various top features which are overlapped on the differential gene expression volcano plot for complete and driver gene data set. So this is how the gene selection workflow went in this study. We started with so many genes. We mapped the probe genes from the microarray data sets into their respective genes. So 20,031 in the complete gene and 919 in the driver gene data set. And then we trained these data sets with various machine learning model and found that 40 and 50 gene features were respectively required for training the most efficient machine learning model. And then we performed differential gene expression, and we found that around 35 genes were common amongst these two data sets. And then we further shortlisted this gene panel using lasso finalized co-aggregation analysis, and we were left with only 8 genes, which were subsequently also used for survival analysis. So these genes in red are downregulated genes, and the one in blue are overexpressed genes. And then we looked at literature and other sources to look for the role of these genes, and we indeed found there a strong correlation with oncogenesis and cancer progression. For example, SCBG is a secretoglobin protein family member, which is significantly downregulated in several cancers, including stomach, cervical, esophageal, and lung cancer. NKX2 is a crucial gene in lung development and morphogenesis, which directly binds and activates the expression of many lung-specific genes, including SCBG1A, B1A1. And selen-BP1 is a member of the selenoprotein family, which covalently binds selenium and mediates its intracellular transport. A decreased selen-BP1 expression is associated with poor prognosis in various cancers. So this was validating our gene panel. Similarly, the other genes are also, which were known to be overexpressed, are indeed related to various processes in cell growth and cell cycle and so on, which are important in cancer progression. So this is, then we took on these 8 genes and generated a risk model. And based on this risk model, we plotted Kaplan-Meier curve to look at the differences in their survival. And we can indeed show that there is a difference between low risk and high risk cancers using this 8 gene panel in the training data set that we use from EU. And then further we validated our finding using an independent data set from TCGA, which was not used in our training. And there also we found strong differences in a panel of 8 genes, gene expression, when they were used to predict on the basis of our risk model. So again, there is a difference between survival of these patients, which show differential expression of these 8 parallel genes. We looked at the various interacting partners of these 8 genes, and we found they were indeed connected to various important processes in the cell growth and cell cycle. So in future research that we'll be using various image analysis for representing various cancers in different clinical settings. And we'll also try to develop algorithms which can be used for personalized treatment. And we all, of course, will work on early detection of cancer, which is a very important challenge in oncology. So this work is already published, and it was mainly done by a PhD student in the lab, Hemant and Anamika. So thank you for your attention. I'll be happy to take any question.
Thank you for the nice presentation. I have 2 questions. The first question is regarding the data set. So the manual control which you have taken, is it the adjacent tissue, or it is totally from the postmortem of?
It is different in different GEO data set, but mostly it is from the adjacent tissue from the same patient.
Same patient. And why have you selected a particular type of lung cancer? Was it, is it the extensivity of the data set for which you selected that?
Mainly availability of the data set.
Availability of the data.
In an ample quantity to be worked with.
And it's very interesting, since you have come up with a set of 8 genes at the end. Do you have any plans? Because these are all leading towards personalized therapy, right?
Right.
So how a patient can be medicated based on the expression of it within that particular patient who needs medication. Do you have any plan to link up these two?
We have to contact the clinical partners to work and validate our findings. We'll look forward to that.
That will be very interesting, because that is where we are going actually.
Yeah.
Thank you.
I think there is a question there. Guys, thank you for your nice presentation, sir. For feature selection, you have to use randomly. We have to select the features, or we have to implement some feature selection algorithm.
So yes, there are various feature selection algorithms. I think we have used recursive feature selection algorithm. So it works on correlation coefficient between various features. So that's how we were using this feature selection.
So it was very nice lecture. I just wanted to know, like you said, you would like to introduce more RNAs into this, because this mRNA is unstable kind of thing. So for the kind of prognosis, or let's say for diagnostic purposes, may not be very suitable. The other thing is how you are going to use this gene panel, because for all those cavity cancers like lung cancer and other cancers, they are mainly detected in a very late stage. So what would be your suggestion to for a person to get it checked, like for these gene panels? How
we have to actually work with clinicians to develop a genotyping panel, maybe, which can be validated in actual clinical samples, and only then it can be introduced to clinical site. So definitely you are right that mRNAs are very unstable, but we can develop probes which will only set of 8 gene probes, which will help us in making a hypothesis that these patients can have a better survival or poor survival, and then it needs to be validated in a clinical setting. Yes, these are just computational finding, which needs to be validated.
So I had a related question. So when you are looking at the different studies, you have addressed a challenge in terms of normalization across these studies. So can you share some details on how you normalize? Because each study could be from a different platform. One could be RNA-seq, another could be microarray. How do you... Because that is one of the... When a new patient comes in and you just use any of these gene panels or other things, if you don't get the normalization right, everything falls apart, right?
Yeah, sorry, I forgot to mention, we use data from same platform. That's why. Sorry.
Okay.
I had a gene question, not related to your talk. Since you're one of these people who are spearheading the Indian Biological Data Research Center, right, in our
IBTC.
Illuminate us about what is installed.
Yeah, I have a small presentation. If we have time, I can just... Yeah. So in a nutshell, this is a DBT mega project where they are funding a Indian Biological Data Center at RCB Faridabad in New Delhi. We have already set up a data center which was funded by DBT, and it's currently having, I think, 5 petabyte of storage. and they have already implemented submission of nucleotide sequences. So whether you submit a sequence at the IBDC portal or submit it in NCBI, the accession number will be in sync with each other. So I will highly recommend that if you are generating data from Indian experiment or Indian funding agencies, you can submit the data in IBDC portal, and which will have an accession number which will be in sync with NCBI, DDBJ and other international databases. So this is arrangement with INDA, which is an international consortium for DNA sequencing. And currently IBDC is also developing a database for metabolomic datasets, proteomic datasets and image datasets, which will be launched later this month. Thank you.
So, Dr. Dinesh, one query there. So, in that repository, will there be an embargo, or the moment you upload it, it's available worldwide?
No, there is a control access platform also. So you can decide when to actually release it.
And that decision stays with the investigator.
Investigate.
Thank
But
you.
in consultation with the committee.
Of course.